Welcome back to another episode of the Found My Fitness podcast. I'm sitting here with Dr. Mark Mattson, who is an adjunct professor of neuroscience at Johns Hopkins University. Mark, I'm so glad to have you here today. This is a long overdue podcast. I've been a huge fan of your research. As a scientist, you have made decades of contributions to our collective understanding of the benefits of biological stress and the supporting evolutionary theory of why almost all organisms actually need stress to thrive. There's a variety of topics that I know you to be an expert on that I'm really excited to talk about with you today, including your understanding the place of plant phytochemicals and our genetic responses to them from a practical and theoretical perspective, your take on how the decades of caloric restriction in animals has panned out, and our attempts to translate those research findings to humans, as well as intermittent fasting as a metabolic switch that has implications for overall health and particularly for brain health. So perhaps to kick things off today, maybe we could start, or maybe you could explain why humans need some biological stress and how maybe modern day society has made that difficult to achieve.
Yeah, during evolution, organisms evolved in environments that were very stressful, even from the simplest of microorganisms like bacteria, where they had to be able to tolerate changes in levels of salinity in the water, exposure to metals that are potentially toxic like iron and selenium, and zinc, and they evolved ways not only that they could resist the toxic effects of these exposures, but actually benefit from them. So, for example, in the case of iron and selenium, we know that now that we need iron and selenium for proper health, but high levels of iron and selenium are actually toxic. And so cells evolve mechanisms where they actually incorporate iron into proteins and use the iron adaptively in ways that help them cope with stress. In the case of selenium, for example, several of the antioxidant enzymes, that is, the proteins in our cells that are able to remove free radicals, those proteins themselves, the antioxidant enzymes, have selenium incorporated into them. So that's one example. And then as we move up the evolutionary tree into multicellular organisms and animals, they evolved in stressful environments. And two of the major stresses are actually food scarcity and predation and competition with other organisms. So individuals that were able to best handle these kinds of stressors, and certainly food scarcity is a stressor, and animals will starve to death if they don't get food. But that stress of the food scarcity is actually a motivating factor, and nervous systems evolved to overcome food scarcity in many different ways. So those are some examples of. And in the case of exercise, individuals whose bodies function well in a food-deprived state and environments where there's potential for predation were those that survived and passed their genes on. So whatever it is those genes did that helped them perform physically well in a food-deprived state had a survival advantage.
We have access to food 24 hours a day, all night. We don't, necessarily need to exercise to get our food either. We can just, get in the car and drive somewhere. We can even have our groceries delivered, to our door. So you've talked a lot about how this constant access to food and not having these periods of food scarcity where people are not eating may have detrimental consequences on overall health?
Yes. One way to look at that is that when we have food available all the time and when we don't have the need to exercise to get through life, our cells become complacent. They do not maintain their ability to cope with the kinds of stressors that cause disease. Oxidative stress is one key example, inflammation. So for a good example is muscle cells and exercise. During the exercise, it's a major stress on the cells in the muscles. There's a big increase in free radical production. There's the cells are electrically active, the muscle cells, so that they can contract. So there's ion fluxes that have to be dealt with. And however, having been exposed to that stress during the exercise, the cells activate gene programs that help them cope with stress and become stronger and more resilient. So, for example, exercise increases antioxidant defenses in muscle cells. It enhances the ability of the muscle cells to clear out damaged proteins, dysfunctional organelles such as mitochondria, which are the energy-producing organelle in the cell. And as well, there are proteins that are initially called heat shock proteins, but their function is to protect other proteins from being damaged. So all of these beneficial mechanisms are stimulated by exercise. So in a person who's sedentary, they have reduced intrinsic antioxidant defenses. They have accumulation of molecular garbage in their cells, accumulation of mitochondria that aren't functioning well, and accumulation of abnormal proteins. And this is also true in brain cells, which is the main thing I study, nerve cells in the brain. There's evidence that's emerging, some from my lab, some from others, that physical exercise, mental exercise, what you and I are doing now around to keeping our mind intellectually engaged, we're right now exercising our nerve cells. They're more electrically active. There's more free radicals being produced in our brain cells right now than there would be if we weren't intellectually engaged. But it's not only okay, it's a good thing, because at the same time, the cells are beefing up their antioxidant defenses, bolstering their mitochondrial function. In fact, we discovered that, and this was originally described by exercise physiologists, and it makes sense. When you exercise regularly, your muscles get bigger, and in the case of endurance muscles, better endurance. And associated with that, there's an increased number of mitochondria, healthy mitochondria, in each muscle cell. So that makes sense. The cells then are more able to generate the ATP to support their function. We find a similar thing in nerve cells when they're active, and most of this is from animal studies, and so we're extrapolating to humans. But in animals, we can look more directly at the brain in kind of an intrusive way. And we find that running wheel exercise, what we call environmental enrichment, where we have the animals in cages where they have essentially like playground type environment where they can maintain their mind more active. And under those conditions, exercise, mental exercise, there's an increase in the number of mitochondria in nerve cells. And associated with that, in some, at least in some brain regions, there can even be an increase in the number of synapses between nerve cells, the connections between the nerve cells. So that's kind of the general thinking, that it's important to keep stressing in a good way, in an evolutionarily conserved way, that is, by stresses that have been normally encountered through millions of years. These transient, short-term, mild, energetic stresses, either the energetic stress of expending a lot of energy during exercise, or the more kind of subtle energetic stress of depriving cells of energy for some extended time period. And I'm sure we're going to talk about what's going on in terms of signaling pathways and, for example, ketones, which are elevated during fasting and during sustained exercise. And you mentioned you talked to Eric Verdin about his work with enzymes called deacetylases, and his work showing that ketones have signaling functions, in fact, gene expression through modulating these enzymes called deacetylases. So fasting does the same thing.
Yeah, maybe we can kind of jump into that. So, there's obviously different types of fasting. There's intermittent fasting, time restricted eating, or even prolonged fasting. Do you want to kind of just maybe briefly describe some of those types of fasting?
Sure. The key thing for your viewers to understand is that when they see intermittent fasting is an eating pattern. It's not a diet. Oftentimes in kind of the lay press and so on, intermittent fasting will be lumped in as a diet, but it's not a diet. It's an eating pattern. A diet is what you eat and how much you eat. Intermittent fasting is an eating pattern that includes intermittent periods of not eating sufficient to deplete the glucose stores in the liver and cause a switch to the use of fats from your fat cells and the ketones produced from those fat cells. So in a person that eats breakfast, lunch, and dinner and doesn't get much exercise, every time they eat, they're replenishing the glucose stores in the liver, and they may never tap in to the fat stores, and therefore their ketone levels will remain low because the metabolic switch hasn't occurred. It typically takes at least 10 hours to deplete the glucose in the liver. So if a person eats breakfast, lunch, and dinner and then has a snack around 8 or 9 o'clock, they may get up and eat breakfast and have not depleted the energy in their liver and have not switched to using fats. So with intermittent fasting, the approaches that have been used in experimental studies, both in animals and humans, are as follows. One is called daily time-restricted eating, where the time window that one eats is compressed into, say, a 6 to 8 hour time period. So that means the person would be fasting for 16 to 18 hours, which is a sufficient time for the metabolic switch to have occurred. And scientists think, based on a lot of data, that this metabolic switching is important for health benefits of intermittent fasting, but also maybe even of exercise in some instances. So daily time-restricted eating is one approach that can result in daily metabolic switching. daily elevation in ketones. So, for example, if a person skips breakfast, eats all their food between noon and 6 p.m., and if they were to measure their ketones, they'll find that in the morning, they'll start to be elevated. Okay. If they wake up and go for a run in the morning, they're already at or in the metabolic switch has already occurred. So they can actually enhance the effect of the fasting in terms of elevation of ketones, and we think in terms of beneficial effects on the brain, the cardiovascular system, perhaps even physical performance, which is an area that's being studied now with intermittent fasting. There's very strong evidence that compared to 3 meals a day plus snacks, intermittent fasting is beneficial for the heart, the brain, glucose regulation, but it's not completely clear yet whether it's beneficial for athletic performance. There's a lot of interest in that. Okay, so let's get back to intermittent fasting eating patterns. Another intermittent fasting eating pattern is one that's now called 5-2 intermittent fasting. And this is where the individual, 2 days a week, they'll only eat one moderate-sized meal of, say, 600 calories those 2 days. Then the other 5 days, they eat normally. And so in that case, 5-2 intermittent fasting, the person will have the metabolic switching occurring 2 days a week, but not the other 5. In fact, there was this 5-2 intermittent fasting, in a sense, kind of triggered the popularization of intermittent fasting. And I'll just take a few minutes to kind of give a historical perspective. We'd done a lot of work in the 1990s and early 2000s showing that intermittent fasting was beneficial for the brains of animals. And we can talk about that, some details on that. And then we'd also published some work on intermittent fasting, reducing resting heart rate and blood pressure, and having anti-inflammatory effects. And then I was approached by a number of clinical investigators. One was Jim Johnson, who worked with asthma patients, and in 2007, we published a small study where we found that these were overweight asthma patients, and we put them on a really rigorous regimen where every other day they only ate 400 calories. So that's not something that can be maintained as a lifestyle, because it's very hard to maintain your body weight with that. But in these overweight asthma patients, over 2 months, it had profound beneficial effects in improving their symptoms, their airflow in their lungs. My lab, we measured indicators of oxidative stress and inflammation in the blood, which went down not right away, but between 2 and 4 weeks of initiating that every other day metabolic switching regimen. Okay, so we published that study. Then I was approached by Michelle Harvey, who's in England and works with women at risk for breast cancer because of their overweight, and they also have a family history. And she came to my lab. She'd seen our work in animals, and there's also some work in animals suggesting that fasting can be beneficial in suppressing cancer growth. So anyway, Michelle and I designed a study where we took these women, 100 women, and we randomly assigned them to either what's now called 5-2 intermittent fasting, or we had a control group where we had them eat breakfast, lunch, and dinner, but each meal had 25% fewer calories than they normally take in. And because we've done a calculation that the long-term calorie intake would be similar in the two groups. The group eating 600 calories 2 days a week versus the group eating 3 meals every day but reducing their calorie intake. Over 6 months, both groups of women lost about 8% of their initial body weight, and both groups had improvements in glucose regulation and other health indicators. But the women on 5-2 intermittent fasting had a greater improvement in insulin sensitivity and lost more belly fat compared to the group that was counting calories, if you will, every meal. Then what happened is a producer at the BBC, Michael Mosley, picked up on that study when we published it in 2010 or 11. And he did a documentary for the BBC, which aired in 2013 or 14. He came to my lab and Walter Longo's lab and Krista Verity's lab. And then, so that aired on the BBC. And then people in the UK got interested in intermittent fasting. And then all of a sudden, like, there's all sorts of things showing up on the internet on intermittent fasting. So it used to be, before the work in the early 2000s, that if you Googled intermittent fasting, the top hits would actually be scientific papers. Now, if you Google intermittent fasting, it's just like a bunch of random people who have some angle on it or so on. But there's now, the good thing about that is there's now a lot of interest from mainstream medicine. And I wrote an article, end of 2019, together with a former postdoc of mine, Rafa de Cabo, a review article on intermittent fasting for the New England Journal of Medicine. And the editors invited us to write the article for two reasons. One, there had accumulated sufficient number of human studies of intermittent fasting, particularly in overweight people, to merit, coverage of it. But the second reason was that many physicians were being approached by patients asking the physicians about intermittent fasting. And in some cases, maybe many cases, the physicians really don't know much about it. They don't know, that there's actually quite a bit of science behind it, both basic and clinical. And from a practical standpoint, they don't know how to prescribe intermittent fasting to a patient and then follow up with them to try to help them switch their eating pattern. It turns out that in the studies, the ones I mentioned, particularly the ones we did with Michelle Harvey in England, but also others, clinical people I've talked to, people, if they can get through the first couple weeks of switching their eating pattern, say 2 weeks to a month, they will no longer be hungry and irritable and maybe can't concentrate well during the time period that they've previously been eating. So, for example, if they decide, okay, I'm going to start skipping breakfast, for the first number of days, even a week or so, they're going to be very hungry, irritable in the morning. And the reason is it takes time for your whole system, everything to adapt to the new eating pattern. And a lot of that has to do with changes in the brain and the neuroendocrine systems that control hunger and the satiety. And we could talk about that. That's a whole another podcast. But one thing we found in pretty much all of our animal studies where we look at the brains, it takes a couple weeks before we see measurable changes in upregulation of antioxidant enzyme levels, increased number of mitochondria in neurons, increased number of synapses, improvements in learning and memory. Okay, so I'm going to stop talking now, Rhonda, and let you ask some more questions.
Oh, well, this is absolutely. I have so many questions. You've brought up so many important and interesting points. But just to kind of speak to what you were just, talking about, this sort of adaptation to perhaps starting an intermittent fasting sort of regimen, and how you adapt to it, and how that a lot of that has to also do with some of the neuroendocrine changes in the satiety and hunger hormones, I guess, is ghrelin and leptin changing.
Yeah.
And is that somehow linked to the production of ketones and this metabolic switching and it happening easier? Or
yes, there is a link with ketones, and it's a very interesting one. And in hindsight, it goes back to the first studies we published with intermittent fasting, which is in rats. This was the one I was at the University of Kentucky in Lexington. So what we did is we took rats and we randomly assigned them to either every other day fasting. And the way that's done is one day you go into the animal's cage, you remove their food completely. The next day you go in, you put back food, as much or more that they want to eat. Then the next day you go back in, you remove all their food. So they're going 24 hours no food, 24 hours food, 24 hours no food. Don Ingram, who's a colleague of mine when I was at the NIA, had shown that every other day fasting can extend lifespan up to 50% in rats when it's initiated when they're young adults. So we took rats and had them either intermittent fasting or not and we did a study where we did like 1 week of intermittent fasting, 2 weeks, and then 3 months. And then we have these models where we can cause damage to nerve cells in brain regions where nerve cells degenerate in Alzheimer's or Parkinson's or with epileptic seizures. And we found that the most striking effect was in the model of epileptic seizures where we administer what's called an excitotoxin. It's a very interesting story. It's a naturally occurring chemical that's produced in algae, and its levels accumulate very high levels during red tide seasons where it's dry. And so there was these incidents an incident in Canada where these people who had eaten shellfish at a restaurant developed memory loss, amnesia. And it turns out that the scientists traced this back to the shellfish. They'd all eaten the shellfish. And then the shellfish had high levels of this excitotoxin. It's called domoic acid. And they had high levels because they were eating the algae that had high levels. So as you go up the food chain, toxins accumulate. Anyway, to make a long story short, the intermittent fasting protected against epileptic seizures and protected the neurons from being excited to death. And we now know that there's a role for ketones in protecting nerve cells against epileptic seizures. In fact, clinicians, neurologists in some cases still prescribe ketogenic diets for patients with epilepsy that don't respond well to the anti-epileptic drugs. Okay, so go ahead.
Well, I was going to ask about whether or not you thought, this metabolic switching, which, you're talking about the metabolism switching from, glucose metabolism to metabolizing fatty acids that are, released from adipose tissue and, in the liver, that produces the ketone bodies, such as beta hydroxybutyrate. If that is something that can occur on a ketogenic diet, what are your thoughts on the benefits of this metabolic switching from intermittent fasting, in terms of, can you get similar benefits by just doing a ketogenic diet, or are there differences between the intermittent fasting and the ketogenic diet?
My opinion is that you can get some, but not all the benefits of intermittent fasting with a ketogenic diet. Early on I talked about how increased activity in neural networks is good for neurons. And with intermittent fasting, there is evidence that during the fasting, there's actually some increase, at least in some neural networks, in activity in neural networks. I guess that kind of makes sense from an evolutionary perspective, that if you're an animal and you haven't been able to get food for a long time, your brain cells better be active. Your brain you better be alert, you better be motivated, and you better be thinking cognitively on, where do I go to find a prey animal or to find fruit on trees, based on so the ketogenic diet will not cause that increase in neural network activity. So, the ketogenic diet, one thing it seems to do is enhance activity of what's called an inhibitory neurotransmitter called GABA. So I'm going to give some Neuroscience 101. Oh, let me ask you, Rhonda. I ask this question to people just out of curiosity, and I'm actually writing a book on this now, second book. Name a neurotransmitter besides GABA.
Okay, just one: dopamine, serotonin, glutamate.
Okay. The third one you named, glutamate is the most important neurotransmitter. A lot of people have heard of dopamine and serotonin because dopamine's links with addiction and serotonin's links with depression. But turns out that at least 90% of the nerve cells in your brain deploy glutamate as a neurotransmitter. And those neurons are distributed in every brain region, throughout the cerebral cortex, other brain regions, hippocampus, brainstem, basal ganglia. Whereas the neurons that produce dopamine and serotonin are few in number, and they're located in discrete regions in the brainstem. They're important, but the only way those neurotransmitters affect behavior is by acting on glutamatergic neurons. Okay, and then the main inhibitory neurotransmitter is GABA, and neurons that deploy GABA are distributed throughout the brain. And their main role is to kind of control excitability of glutamatergic neurons throughout the brain. So if you were to block the function of GABA producing neurons, your brain circuits would fry themselves. On the other hand, if you have too much GABA, then it can quiet down the glutamatergic activity so much that your brain doesn't function well. So an example of that is drugs called benzodiazepines, like Valium. they activate GABA receptors, so they quiet down the glutamatergic neurons. But if you take too much Valium, if you were an animal in the wild and you hadn't gotten food in a long time, you don't want to take Valium. You want your nerve cell circuits to function. Anyway, so the intermittent fasting, similar to intermittent exercise, you get this activation of neural networks. One of the main effects that I haven't even talked about yet in the brain of exercise, intermittent fasting, and intellectual engagement is to increase the production of proteins that are called nerve cell growth factors or neurotrophic factors. One of those proteins that's produced in response to activity in neural networks and in response to the metabolic stresses of exercise and fasting is called, that neurotrophic factor is called BDNF, brain-derived neurotrophic factor. It is the most heavily studied such nerve cell growth factor in the brain. And it is essential for learning and memory. You can't knock out the BDNF gene in mice. They'll die. You can reduce levels genetically, and then that will impair learning and memory. Actually, interestingly, if you reduce levels of BDNF by about 50%, the animals will overeat and become obese. So BDNF is also involved in regulating appetite. Okay, so let's see, where were we? Right, so ketogenic diet, it will definitely, your neurons will switch from using glucose to ketones. We think that's good because essentially ketones is a more efficient energy source for cells than glucose. And there's less free radicals generated in the burning of ketones compared to glucose. And then the ketones have these signaling functions affecting gene expression that glucose doesn't have. So I guess two things. The ketones are a good energy source for neurons, and they have signaling functions, but those signaling functions are more limited than are activated by exercise or intermittent fasting or keeping your brain active.
So you mentioned about, the different types of metabolic stress, whether that be exercise or intermittent fasting, and how, there's an increase in BDNF in the brain. In animal studies, and I kind of want to go back to seeing, what your thoughts are on translating, these animal studies to humans. Obviously, animals have very different metabolic rates than humans. For example, if you were to, fast a rodent for 20 or 48 hours, they lose 20% of their body weight, whereas a human would only lose 2% of their body weight from a 24 or 48-hour fast. But I also want to ask you, have you seen additive effects in your studies on animals, or have you looked at additive effects of animals that are constantly exercising or routinely, so a physically active animal with their little exercise wheel, but you also expose them to these periods of food scarcity, intermittent fasting? Are the two combined better than just one type of metabolic stress?
There is evidence that we've obtained that a combination of exercise and intermittent fasting can be better. So I'll give you two examples that we published. One was we published a long time ago, a former graduate student, Alexis Stranahan, and it's actually relatively simple to study that design, but we had normal mice and we had mice that have type 2 diabetes. And they have type 2 diabetes because of a genetic, they're genetically engineered to have a defective leptin receptor. Leptin is a hormone that when you eat a meal and your stomach gets full, it's released into the blood and it travels up to your brain, a region called the hypothalamus, and essentially tells you you're full, stop eating. So these diabetic mice, leptin receptor mutant mice, they don't get the stop eating signal, so they're presumably always hungry. Okay, so then what Alexis did is she divided these normal mice and diabetic mice into 4 groups. The normal laboratory conditions, which is they're sedentary in relatively small cages, no running wheels. And then they're fed ad libitum. They can eat as much as they want every day. Then she had a third group that were fed ad libitum and they had running wheels in the cages. And then the fourth group, wait a minute, what did I say? Yeah, sorry, I think I'm interested. So she had ad libitum, sedentary, ad libitum, running wheels. Then she had every other day fasting, sedentary, every other day fasting, running wheels. Then she let them go for 3 months. And she took out their brains, and there's a brain region called the hippocampus, which is in many respects the most intensively studied brain region for several reasons. One, it's critical for learning and memory. It's kind of all the information from our eyes, our ears, our other senses funnels into the hippocampus, and that's where the initial, if you will, associations between a sight and a sound, a bear jumping out on the trail and growling, you hear its growl. The next time you go walking on the trail, you hear a bear growl. You don't have to see it to, have a mental image of a bear. So the hippocampus is critical for these initial associations. And then a second reason the hippocampus is heavily studied is it's a focus of dysfunction and pathology in Alzheimer's disease, epileptic seizures, even stroke in some instances. And a third reason is it's easy to study because the circuitry is actually relatively simple compared to the cortex. So we can put electrodes in, we put electrodes in the glutamatergic neuron here, and we stimulate a glutamatergic neuron down here, and we can record that. We can activate a GABAergic neuron and record reduction in activity. So anyway, what Alexis found was that first, what she did is she measured the number of synapses along the dendrites, the part of the neuron that receives information coming from another neuron. So she simply counted, not simply, it actually takes a lot of work, but counted synapses and found that 2 things. One, the diabetic mice, regardless of whether they were intermittent fasting or had running wheels in their cages, had smaller number of synapses than did the normal mice. And then she found that the running wheel exercise and the intermittent fasting increased the number of synapses, and the combination of intermittent fasting plus running wheel got a further increase in the number of synapses. Okay, so that suggests that intermittent fasting can enhance the effects of running on increasing number of synapses between neurons. And then she took the other hippocampus from the brain and measured BDNF levels. And I mentioned BDNF is important for learning and memory. It's actually also important for formation of new synapses. And she found that the intermittent fasting and the exercise, each alone increased BDNF levels, and the combination got a further boost to BDNF levels. And then, but she did not find that in the diabetic mice, synapse levels came up to the level of normal mice. So there was some beneficial effect, but you couldn't get it back to like they had never been diabetic. Okay, so that's one example. Then the second example has to do with endurance exercise. And this work was done by former postdoc Christina Morosi and a postdoc fellow, Keelan. I'm actually looking at a... So this, can you see this?
Yes.
Can you see the whole picture?
I can. Mm-hmm. Yeah.
So this is my running group from the lab about 2016 or something. Anyway, so the work I'm going to talk about now was done by Keelan, Mole, and Christina Morosi. Christina's not on here. She wasn't fast enough to make our A team. So, so what they did is they took mice and they had 4 groups: ad libitum, sedentary. These are all normal mice. Ad libitum, sedentary. Ad libitum, daily treadmill training for 45 minutes every day for 2 months. Then she had a group every other day fasting, no treadmill training. And then the final group was daily treadmill training while they're on every other day fasting for 2 months. And then at the end of the 2 months, they did a maximum endurance test. So 45 minutes a day on the treadmill. And then every week they increased either the speed of the treadmill or the incline. So they're actually, increasing the work over the 2 months. So they did maximum endurance and test how long can the mouse stay on the treadmill without giving up. And they found that, as you'd expect, the animals that had not done treadmill training had much poor endurance than the animals that did treadmill training, regardless of whether they were on intermittent fasting or not. But there was a statistically significantly better endurance in the mice that were on intermittent fasting during the 2 months of treadmill training. And they measured ketones, which were elevated with intermittent fasting, and the exercise increased the ketone levels much more, almost twice as much when you take their blood after the end of the treadmill training. And then they took out, they did a lot of analysis of the blood. They did something called metabolomics. And then they took out muscle cells from the soleus muscle, which is an endurance muscle. in the leg of the mice. And they did some measurements that suggested that intermittent fasting and the exercise increased the number of mitochondria in the muscle cells, and that the increase was the most when you have the combination of the intermittent fasting and the exercise.
Very cool. I remember reading a meta-analysis a few years back looking at human studies, people that exercised either fasted or in a fed state, which unfortunately there's a bit of a confounder, because if you look at the dietary composition of most of the foods these athletes were eating before exercising, they're very high in carbohydrates, and oftentimes even refined carbohydrate, where you have like some toast and jam or something like that. But regardless, there was a variety of metabolic adaptations that, for example, mitochondrial fatty acid oxidation was enhanced, mitochondrial biogenesis. These things were somewhat blunted in people that exercised in a fed state, whereas if they had done it in a fasted state, there was these adaptations were occurring even throughout the day when the people were not exercising. And so there was also some effects on there were some if people had exercised fasted, but they were doing a very, very long sort of endurance training type of activity, then there's some performance issues, weren't as good as if they had exercised while in the fed state. But since reading that meta-analysis, I've tried, for the most part, I'll do a 3-mile run, or I'll get on my Peloton and do a 20-minute high intensity, Tabata or something training. And I do it fasted, and I definitely have adapted to it over time. But I do it because I'm thinking to myself, well, I want those mitochondrial adaptations. So, so it's really interesting that your experiment that you mentioned.
One point you just mentioned is important, and this can occasionally get lost as these conversations go on, and that is, it's a switching back and forth intermittently is important. The number, for example, in the muscle cells with exercise, the number of mitochondria doesn't increase during the exercise. It increases during the rest period. But if you had never exercised, you would have never gotten the stimulus that triggers what's called mitochondrial biogenesis, the increase in number of mitochondria. So they're switching back and forth between the metabolic challenge, whether it's fasting or exercise, and the recovery, whether eating, resting, sleeping. And there's definitely a limit to all of this. Obviously, with fasting, starvation, if you start to lose muscle mass, then you're fasting too much. And as you mentioned, you can overdo it with exercise too, and sometimes get diminishing returns. So it's important to have these recovery periods.
You've talked about the importance of what are called the refeeding phase. Both you and Dr. Walter Longo have discussed this in publications, and Walter on the podcast previously. And it's a really, like you mentioned, it's a point that a lot of people don't focus on, that recovery period. How important, for the refeeding period, how important is that, and how long is that window? Do we know? Is it like a week? Is it just a couple of days?
Well, with these intermittent fasting eating patterns I'm talking about are kind of trivial from an evolutionary perspective. That is, limiting the time window you eat to 6 to 8 hours each day. The remaining, 16 to 18 hours is more than sufficient. to recover. If you tried to do fasting for 5 days, 1 day recovery, fast another 5 days, 1 day recovery, and keep that up, it won't be long before you're going to start to have problems. So, and it's the same with exercise. It's, there's, my understanding is there's quite a few ultra marathoners who, when they get in their 50s and 60s, start to have a lot of problems. So there could be some, long-term consequences of overdoing it, whether it's with fasting or exercise. And what you and I are talking about today is well within any bounds of even getting close to having adverse effects and having to worry about, am I recovering or not?
But perhaps more of someone doing more of a prolonged fast might have to consider, the refeeding and how important.
Yeah,
I know.
In Europe, for example, in Germany, there are clinics where people go in for a couple weeks and they'll fast for 10 days to 2 weeks under, supervised. It's kind of like a resort, actually, and everybody else there is doing the same thing. And they're collecting a lot of data and starting to publish, and but they see a lot of improvements in health indicators even within that 2-week fasting period, but they don't have really good data on long-term effects. And in talking to the people who run these centers, oftentimes a person will only do this once a year, and so they may be overweight and have insulin resistance, and they'll go in and they'll show some, even during that short period, a little bit of improvement in their insulin sensitivity, but then they'll come back a year later and they're back where they were or worse. So one question then is, if someone wants to fast for longer time periods, what frequency would be reasonable for long-term health? You've interviewed Walter Longo before, and he's done a lot of work with this eating pattern where the subjects 5 consecutive days a month, they'll eat only one moderate size meal, and then the other days of the month eat normally, and that seems to be beneficial. The key thing is, how long can people maintain this in their lifestyle, incorporate it into their lifestyle? And daily time restricted eating, for example, seems to be easy for a lot of people to do for years and years and years. Maybe 5 days a month, that's perhaps. But at some point, you have to think about it that way, looking kind of a long view of what can you. And then another thing is, what about, so daily time restricted eating, if you skip breakfast, then you can still have lunch with people at work and dinner with people. Whereas if you're doing something else, maybe like Walter's, maybe 5 days a month, you're not going to be able to, you may go out to dinner with somebody and say, well, I can't eat anything today. So, yeah.
Right. It's a lot more challenging.
One thing I want to get into, and you mentioned very early on in the introduction, was, and I think we're getting there, is diet composition and the question of there's good evidence. Yes, eating vegetables and fruits is good for health. And why is that? And the emerging evidence that has kind of turned the load up on antioxidant idea on its head. And so, yeah. So during evolution, it was we're omnivores, and the same would be true with other animals that are omnivores or herbivores. It is advantageous to be able to eat plant materials, fruits, nuts, roots, leaves, because they have energy. And however, it's not necessarily advantageous to the plants that we eat them. In fact, it's generally not. And we know that in the case of insects, but it's also true with herbivores and so on. So most plants, the most vital parts of the plants in terms of propagation of the species, if you were to eat those, they have a bitter taste. The skin, so for example, the skin of grapes or apples, and broccoli, seed producing part of the broccoli, and so on, they have a bitter taste. And so the thinking is that it was to our advantage to be able to eat these, even though they have a bitter taste and have these naturally occurring pesticides, is what they are. The plants are producing their own pesticides. So that's, in fact, one of the reasons plants produce so many of the chemicals is that many of those chemicals, their function is to dissuade insects and other organisms from eating them. That's their main function. Okay, so we co-evolved with these plants. And as we co-evolved with them, we evolved several ways to protect ourselves from overdosing on these pesticides that the plants normally produce. So one way is bitter taste. The second way is vomiting. The third way, which is interesting from human environmental health standpoint, is we have evolved enzymes in our liver that rapidly remove these potentially toxic chemicals when we eat them. They're called cytochrome P450s. Okay, but the fourth way is that the individual cells in our body have evolved to respond to some of these chemicals by, for example, enhancing their antioxidant defenses, or enhancing their ability to even extrude the chemicals. So I'll give you a few specific examples. I mentioned broccoli. Many of your viewers, probably if they're into health, they've probably heard of sulforaphane, which is a chemical that's in broccoli, and there's quite a bit of evidence that it can be good for health. And one of the ways it is it activates antioxidant defenses in our cells. So it's similar to exercise and fasting, which are stressors. The stress of this chemical that's in the broccoli that we consume, the stressor is the chemical. It's activating antioxidant defenses. Another example is curcumin, which is in turmeric root, which in Indian food is very common. And it also activates antioxidant defenses. The most commonly consumed plant toxin by your viewers, very likely, is caffeine. So if you were to take concentrated caffeine and put it on your tongue, it has extremely bitter taste. It's possible to overdose and die from caffeine, and there have been some documented cases of actually people essentially eating caffeine powder. If you take tea leaves, coffee beans, even ground coffee beans, put them on your kitchen table in the summer, assuming you don't have an ant-free house, and there's ants crawling on the table, you'll notice that they avoid the coffee and tea leaves. And they're avoiding caffeine and other naturally occurring good toxins in them. So that contrasts with sugar or whatever, any a lot of other highly palatable, addictive foods. So anyway, I was invited to write an article for Scientific American on this general notion that the reason fruits and vegetables are good for our health is not because there are free radical scavenging things like vitamin E or C can help in certain ways getting rid of free radicals. That's not the answer. The answer isn't that we want to swamp ourselves with things that sop up free radicals, because actually free radicals are important in our cells for normal signaling and in our adaptive responses to stress. Instead, the chemicals that are good for health seem to be acting by triggering mild adaptive stress responses in our cells that overlap quite a bit with exercise and fasting. And these chemicals, again, they're concentrated in the exposed parts of the fruits, like the skin of the fruits. Here's a really interesting thing: green tomatoes. All right, so bugs usually don't eat the tomatoes till they start to turn red in the garden. Okay, so why is that? The reason is they like the taste of the red ones better than the green ones. So it turns out there's this chemical that's been called tomatidine that's highly concentrated in the green tomatoes, and then levels of tomatidine decline dramatically as the tomato turns red. Okay, by the time the tomato starts turning red, the seeds in the tomato that have the potential to give rise to another tomato plant and propagate the species, they're ready to go. So I guess mainly this applies to birds, but so say birds won't eat green tomatoes either, so let's go with that. So the bird eats the red tomato, and they may eat some of the seeds. The seeds can resist going through their digestive system. Then they poop out the seeds, and the seeds can grow. And they may poop out the seeds, whatever, half a mile away. So now the tomato plants are, and that's, so these are all evolutionary mechanisms that give advantages to one or both of the species during their coevolution.
If a person is exercising regularly and also doing some form of intermittent fasting and they're getting these activation of some of these stress response pathways and the beneficial effects, and you're getting the metabolic switching. And is there any need to also take in these plant phytochemicals that are also activating, perhaps the same or different or both, stress response pathways? In other words, like, can you just say, well, I don't need the to eat the plant compounds because I'm exercising and I'm doing intermittent fasting?
I wouldn't do that or say that. What hasn't been done though, Rhonda, is there, and the reason is it's expensive. There haven't been studies where within a study they do all these various combinations, of, okay, we're going to do this study with, we'll have some people intermittent fast, we'll have some exercise, we'll have some do both, we'll have some, whatever, take in sulforaphane, and then we'll have them do sulforaphane plus intermittent fasting and so on and so on. And my view on diet composition is one that I think makes sense from data from epidemiological studies, the blue zone, where there's certain places where people have exceptional longevity, and all of them have diets that are mostly plant-based, and then the evidence that simple sugar is bad for health is overwhelming. The saturated fat, I think the evidence is pretty strong. It's better to eat fish than red meat. And so, complex carbohydrates are better than simple. I think that, people shouldn't say, well, I can just eat at McDonald's every day if I exercise and intermittent fast, because they would be better off than having been intermittent fasting and exercising. If they're going to eat at McDonald's and don't want to give that up, then exercise and intermittent fasting will counteract that somewhat, but it's still kind of stupid.
The way I also think about it is, you have something like sulforaphane, which we've talked quite a bit about on the podcast. I've interviewed.
Yeah, I know. Jed, he's at Hopkins.
Yeah, he's a good friend of mine. And so, you have something like sulforaphane that's activating the NRF2 pathway, which has, all these downstream effects on phase 2 detoxification enzymes, activating those and inhibiting the phase 1 biotransformation enzymes, which are involved in potentially making a pro-carcinogen, an actual carcinogen, and there's all these pathways that, I don't know how much exercise or intermittent fasting, activates those pathways. I'm sure to some degree, but, it seems as though different types of stressors do have a more robust effect on certain types of stress response pathways.
Yes.
And so why wouldn't you want to diversify?
Yeah, that makes sense. Yeah. Although I should say that, so go back to sulforaphane and NRF2 ARE pathway. That is activated by exercise and fasting. And so I would say these chemicals that are in the plants are more targeted in what they do. Compared to exercise and fasting, those chemicals affect a more limited number of pathways. In other words, you can't just take sulforaphane and it's going to substitute for exercise or intermittent fasting.
Right. Kind of shifting back just for a moment to the fasting topic. We've talked a lot about metabolically unhealthy people with type 2 diabetes, or perhaps obesity, or animal models of such, and the benefits of intermittent fasting in those people or animals. How much can a healthy person who perhaps is not at risk for obesity or metabolic syndrome benefit from intermittent fasting?
Yeah, that's a complicated answer. I'll expand a little bit. How much and in what ways? There have been far fewer studies in humans of intermittent fasting on normal weight, healthy people. In animals, the control group is always fed ad libitum and they're sedentary. So really in the animal studies, the control group is couch potatoes. And that's why, we look at, I would say in extrapolating the animal data to humans, I'm very comfortable in saying intermittent fasting will have very clear, measurable, highly significant benefits for overweight people. But, since all the animal studies, it's the control group is couch potatoes, and we're seeing good effects on the couch potatoes, we can't say, does that apply to humans or normal weight? What there have been studies in normal weight, healthy humans looking at some health indicators, blood glucose levels and insulin, and looking at ability to maintain and build muscle mass. So resistance training studies, several published studies where they found that with daily time restricted eating, people were able to maintain and build muscle just as well as people who weren't on daily time restricted eating. But it's undoubtedly the magnitude of any beneficial effects on health in people who already have a healthy body weight are going to be less than in people who are overweight. And the effects on improving glucose regulation in people with type 2 diabetes or already have insulin resistance are going to be quantitatively much greater than in people who have, already have normal insulin sensitivity. So, I would say that there will be some benefits, but it's going to be quantitatively less. Does that make
Quantitatively
sense?
difficult. It does, difficult to even measure, because as you've been discussing throughout the podcast, the benefits of metabolic switching, the production of ketone bodies like beta hydroxybutyrate, and their signaling effects on BDNF, increasing BDNF, and also, producing less reactive oxygen species because they're, more energetically favorable or, used easier by the mitochondria. It seems as though activating autophagy, as you mentioned, the clearing away of, damaged stuff and organelles and things within a cell, those, sorts of effects happening, even if they're not if the magnitude isn't as great, it seems as though that has to be beneficial long term in terms of activating stress response pathways and they're going to help you deal with the stresses of aging better.
Yeah, I agree.
Do you think that? Sorry.
Go ahead.
No, go ahead.
One issue is with, one thing that's being found is it's important to maintain muscle mass as you age. So I think people need to be a little bit careful, to take in sufficient energy to maintain their muscle mass. So, for example, the people in the Calorie Restriction Society may be overdoing it, and the story of Roy Walford, who was in Biosphere 2.
You can tell the story.
Okay. I can't remember exactly when it was, 70s or 80s or 1980s. I can't remember. There's something called biosphere where these two people down in Arizona, essentially they built this bubble and they grew plants in there. And the goal was see if they could be self-sustaining for long term. And so what happened was they were all right for a while, and then after a while they started not being able to generate sufficient food, and they lost a lot of body weight, and essentially on the point of starvation. And so they had to finally come out of the biosphere. And one of them was Roy Walford, who had also done a lot of important work on calorie restriction. Actually, a lot of very important early work with Rick Weindrich at University of Wisconsin, showing that calorie restriction can extend lifespan. And then Roy Walford, when was it? So as you get older, your perception of time is messed up. Roy Walford died in, I'd like to say, like 15 years ago or something. ALS, Lou Gehrig's disease. So it's a muscle wasting disease involving degeneration of the neurons that innervate those muscles. And that was really interesting because I mentioned these studies that we'd done with intermittent fasting in animal models of Alzheimer's, Parkinson's, epilepsy, and we found it was beneficial. But we also tried every other day fasting in a mouse model of ALS. And what we found is it made it worse. So, of course, these mice are destined to have ALS. So the kind of the abnormal processes are already in motion when we put the animals on intermittent fasting, but their motor function, we have ways of testing their motor function, declined much more precipitously when they were on intermittent fasting. So I think that's the important. And then my own personal case. So I've always had a low BMI, and I started doing daily time restricted eating like 30 years ago, not eating breakfast. And I was kind of an endurance semi-athlete, trail running, mountain bike riding. Actually, when I was a kid, I used to race motocross. But anyway, I did a lot of running. And so my BMI is always around, it's very low, like 18.5, somewhere there. And then 2 years ago, I had a mountain bike accident, and not to be too gory, but essentially tore my rectus abdominis muscle off my pubic bone and had adductor partially coming off. I had to have 3 surgeries, and I've had other issues now. But so I've lost muscle mass, particularly in my legs, during this ordeal, and I'm having trouble building it back up. So, like, in hindsight's always 20/20, and I couldn't have predicted I'd have a mountain bike accident and have all this going on. But in hindsight, I probably would have been better off. And I didn't really do any resistance training because when I started running in the 70s, there was no such thing as cross training. It was like the training is you run, you stretch and you run. And somehow, I guess because I'm stupid, I didn't pick up on, hey, as people started doing cross training stuff, I didn't pick up on, hey, I should be working my core and I should be, keeping, some muscle mass. So all these years I was mainly just doing the same thing. But anyway, so that's just like a personal anecdote. And, I guess what I'm saying is having a low body weight can be good, but you have to be a little careful during aging to make sure that you have a good muscle mass as part of that. Like the main, well, I guess I was mostly muscle mass anyway, but still, yeah, more muscle mass.
It brings up a good point on some of the, safety concerns with practicing intermittent fasting and certainly prolonged fasting, and, whether or not elderly people, someone over the age of 70, can do, a 5-2 type of fast, or certain, even, daily time restricted eating. And maybe they should be, doing that along with resistance training, if they even should do it. Do you have any thoughts on that?
My thoughts are that exercise and a healthy diet would be more important for them. Yeah. And, if they're, we don't know. The answer is we don't know. There have not been, there needs to be studies in elderly people of intermittent fasting starting it in the elderly. So it's just not clear. So right now, the main focus, at least from mainstream medicine, is on people who are overweight or with insulin resistance. There is some interest in also people at risk for cancers, because there's really strong, as you know, being overweight is a risk factor for a lot of different types of cancer in both men and women. So in as much as intermittent fasting can keep your body weight down, that's good. But there's also evidence that intermittent fasting can actually, in animals, it definitely suppresses the formation of tumors, spontaneous formation of tumors, and it can greatly reduce the growth of cancer cells implanted in the animals, and it can enhance the killing of cancer cells by chemotherapy, drugs, and radiation. But anyway, so elderly people, if they have good muscle mass and are physically active, I don't know, they can try intermittent fasting. If they have the same overall calorie intake, then they're not going to lose body weight, whether they're eating all their food within a 8-hour time window or not, or 2 days a week, if they make up for the calories. Then another group would be little kids that are growing fast. Children who are overweight with obesity, maybe there's interest by pediatricians in this, and I've had several contact me, one up in New York, who, what he's had some success with is kids with obesity. He gets his parents to buy into it, and then gets both the parents and the kid to switch their eating pattern. And he's had success in helping some of these kids get their body weight down by switching their eating pattern to intermittent fasting. So women with obesity and type 2 diabetes, there's increased risk of autism, or having a child that is on the autism spectrum disorder, compared to normal weight, healthy women. And that's very interesting. And I can send, I don't know, you can put stuff up on your website, Rhonda, right?
Absolutely.
So I don't need a website. I just go through you. So I'll send you a few articles that you could post on your website. Is that okay?
Yeah, and we also post them on the video. We put the figures and study titles
Oh, okay.
and information. So whatever you're
Anyway,
talking
I'll
about,
send
we're
you
going to
this one
find.
on autism. So when one looks at, so we know that in the 1970s and 80s, autism was kind of, a lot of people never heard what's autism. What's autism? I didn't. When I went to high school, I never heard anything at school or from my parents about autism. And then beginning in around the late 80s, 90s, and then more so, there's an increased incidence of autism. Some of that's due to increased recognition that the kids are having trouble concentrating, they're avoiding social interactions, and so on. But that doesn't seem to explain all of the increase. It turns out there's a nice, if you track the increased incidence of autism and the increase in maternal obesity and type 2 diabetes, it tracks really well, through the, from very little maternal obesity in the 70s and 80s, and then increase autism. Then, so the neuroscientists have good evidence that in autism, during brain development in the embryo and the uterus, the brain grows more rapidly than normal. And probably because the mTOR pathway is like just, the person's, if they have obesity, type 2 diabetes, they're undoubtedly not exercising or, calorie restricted. And so they never have the metabolic switching. mTOR pathway, which is the growth mode, is on. And so nerve cells, the neural stem cells proliferate more rapidly. Neurons grow more rapidly. Connections start forming. And then what happens is there seems to be hyperexcitability of neural networks. And this has been documented by doing what's called functional magnetic resonance imaging in kids with autism. Kids with autism have a huge increase in incidence of seizures. Now, I should say, not all kids with autism are born to women with obesity or type 2 diabetes. Not all kids with autism have seizures, but there's a big increase incidence. That is, more kids with autism have seizures than kids that don't have autism. And then in animal studies, there's some genetic models that are actually pretty good for autism. There's something called Fragile X syndrome, and the gene is known for that. And when that gene abnormality is put in mice, and then you put the mouse in a cage with a bunch of other mice, the mouse goes in the corner and doesn't want to interact with the other mice. So you can kind of measure this propensity to interact, so kind of social withdrawal. And those mice in their brains, they have hyperactivity of glutamatergic neuronal networks. So there is some evidence that kids with autism, exercise can help them. Certainly I used to coach high school cross country and my kids ran. And I know that during the cross country season, the kids their mood is much better than when they're not running. They're more relaxed, less ornery. And right. And then the exercise and intermittent fasting upregulate the GABA tone. And ketogenic diets will do that too. So it'll be interesting. I think it would be interesting to try, intermittent fasting in kids with autism. No.
Or perhaps a ketogenic diet as well. Very, very interesting, Mark. To kind of shift back to the women part of this story, there's been a lot of questions about whether or not fasting affects women differently, different than men, and if women, if women, should fast, or if it affects their cycle, menstrual cycle, or hormones like thyroid, things like that. Whether we're talking about like a intermittent fast that's more strong, like longer than time restricted eating, something more like a maybe 24 to 48 hour, or perhaps even a more prolonged fast. Do you have any thoughts about that?
Yeah. Major calorie restriction. So I'll start with animals, then go to humans. So in animals, for example, I had one postdoc, Bronwyn Martin, in that, like 2005 or something. She took rats and she put them on ad libitum feeding, 20% daily calorie restriction, 40% daily calorie restriction, which is a lot, or every other day fasting. And she had both males and females. So, and then she followed them over time. And in the females, she essentially did vaginal swab and to do staging of the cycle. And so as far as that goes, the rats with 20% daily calorie restriction, there was no change in their estrous cycles. The rats with 40% calorie restriction, and this was over a period of like 4 or 6 months, they shut down. They stopped cycling. And they lost a lot of body fat over those months. Then the rats on every other day fasting, they kept cycling, but there was some increase in irregularity of the timing between the cycles, but they were still presumably fertile. Then what else? She did all sorts of stuff. She tested their learning and memory. Oh, then interestingly, the males, even on the 40% daily calorie restriction, so the males, their sperm count didn't change, and they didn't lose as much body weight as the females over these months. So then, and then she looked at the activity of the rats moving around the cage, and the females, when they're on major calorie restriction, became very active. Like they're moving around the cage a lot, looking for food, maybe. So my interpretation of this, and we had like a few sentences in the discussion of the article. Okay, in the wild, if there's animals are getting to the point of starvation, so that would be the 40% calorie restriction. The females, they don't want to get pregnant. If they get pregnant, there's no food to support, development of their baby. So they shut down their cycling. They become more active looking for food. The males, the males, before the males starve to death, it would be advantageous to them to be able to inseminate as many females as possible before they die of starvation. So they stay fertile. So that's my interpretation of that. The female has the egg that's the potential passing the genes on. And, so I don't know. But with the intermittent fasting, they kept cycling, and their activity in the cage increased a little bit, but they maintained pretty good body fat compared to the 40% calorie restriction. One issue is if and this applies mainly to adolescent girls. If an adolescent girl goes to intermittent fasting eating pattern, would she be more prone to developing anorexia nervosa, which is a kind of a obsessive compulsive like psychiatric disorder? The answer is we don't know. We just don't know. So from an evolutionary perspective, you would think that would be selected against, but so, and it's not clear. I guess I don't know enough about anorexia nervosa, but how back in human recorded history, how is anorexia nervosa even common, or is this something that has arisen more as girls are more conscious of their body image and so on, and therefore there's this psychological factor that I don't know if that's something that, I guess what I'm saying, it doesn't make sense to me that anorexia nervosa would be something that would not be strongly selected against during evolution. Right, because these girls often, they usually quit cycling too. So yeah, I guess we just don't know. On the one hand, intuitively, it'd say it wouldn't be a good idea to recommend this to adolescent girls, but if they're with obesity or overweight, I don't know. We just don't know. I don't know. It seems like it may be okay, but we just don't know.
You're talking about with adolescent
Yeah.
girls. Well, so what about women that are not adolescent and are, do not have an eating disorder and are perhaps even normal weight, not obese or overweight? Is there a concern with other hormonal imbalances? I don't even know necessarily, is it a bad thing? I mean, if you're amenorrheic for a short period of time and you go back to eating normal calories, what does that mean? Do you delay your reproductive lifespan longer, or is there even implications that?
Well, in animals, that's what happens. For example, if we were to take these rats and do 40% calorie restriction for 3 months or 4 months, so that they stop cycling, and then you put them back ad libitum feeding, they gain their body weight back, they start cycling. These aren't our studies, these are other studies. Then those rats will be able to keep cycling to an older age than they would have previously stopped cycling. So in other words, in theory, maybe you could extend age of menopause by shutting down cycling for 10 years. This is like just
Speculation.
This is just thinking and not anything that's approaching it, coming close to even encouraging someone to do something like that, which would be crazy. But it's an interesting thing to think about. I don't know there haven't been studies where this is a big thing that's lacking in this field is studying hormones, except for like simple things like leptin and ghrelin. So, for example, FSH, LH, oxytocin, anything produced. Oh, we do know in animals anyway, there seems to be increased activation of the hypothalamic pituitary adrenal stress response system. And that's the system that results in increased levels of cortisol. And this was something that was noted early on in the animal studies. The animals live longer when they're on calorie restriction or intermittent fasting, but they have elevated cortisol levels, which is usually, in the clinical arena, that's not a good thing because it can suppress the immune system. However, so the animals are living longer. And we did a study, Jaewon Lee, who was a graduate student when I was at Kentucky, and then he came to Baltimore with me when he moved. So this gets a little bit into endocrinology. Cortisol, there's two receptors for cortisol, two proteins inside the cell that bind to cortisol, and those cortisol binding proteins are transcription factors. So cortisol comes from the blood into your cells. It could be a muscle cell, nerve cell, doesn't matter. And they bind to the receptor, and then, which is a transcription factor. It then goes into the nucleus and affects the expression of certain genes. And in fact, the Nobel Prize was given to the person who, I'm blanking on the name. I should know this, who discovered this. Okay, so I mentioned there's two receptors for cortisol. One is called the glucocorticoid receptor, or GR. The other is called the mineralocorticoid receptor, or MR. So there's been a lot of studies on cortisol in relation to chronic, uncontrollable psychosocial stress. So people who are, whether it's, their work or life situation, they're chronically stressed out, they have elevated cortisol levels. And it's been shown that in that case, in the brain, nerve cells in the brain have a decreased level of one of the cortisol receptors, the MR, and an increase in GR. So the way that the cells are responding to the cortisol is changing, not just the cortisol levels. So we did a study where we measured levels of GR and MR, the two different receptors for cortisol, in the hippocampus of mice that had been on every other day fasting or ad libitum control feeding. And what we found is that in contrast to chronic uncontrollable stress, the intermittent fasting caused a decrease in levels of GR and a sustained level of MR. So the take-home message is there's increased activation of stress response pathways with intermittent fasting, but the ways your cells respond to the stress is different than the bad ways your cells respond to bad types of stress, chronic uncontrollable stress.
That's really important. That's a very important point to make. Just going back to one thing you were talking about, comparing your severely caloric restricted animals to like alternate day fasting. I think that's a really important point to distinguish because, in the context of what we were talking about with women's cycle, there wasn't really, much of an effect on the cycle, alternate day fasting, which, by the way, in rodents is a much stronger, like a woman doing a 24-hour fasting and then, alternate day would be probably much more significant of a fast in rodents, right?
Yeah.
But anyways, the differences between and uncoupling the benefits and, or just
wait a minute. I think you said it backwards. So it's a more significant a fast in the rodents.
Yes, it's a more significant. Yeah. Exactly. Okay. Yes. Okay. Thank you. Yeah. 24-hour fast in rodent would be, I'm not even sure, would it be like 72-hour or something in humans? Much longer.
It's more. Yeah, much more. That, as you mentioned, mice, they die if they don't have food. If you go beyond 3 days or so, they will die. So that's kind of, so 1 day is a third of that, right? So I would say, a human can live, it depends on their initial body fat, but they can live maybe 2 months.
That's a big
without
difference.
Food. So I'd say, a day complete fasting could be equivalent to no, it can't be that much. Maybe
5 days.
I don't know. Anyway.
Yeah. Right. Well, you know what? I don't know if this makes sense to you, Mark, but if you look at some of the work from, for example, Valter Longo, when they've looked at a 48-hour fast and the drop in IGF-1 levels goes to about 50%. And in humans, looking at what it takes to drop IGF-1 to 50%, it's anywhere between 5 to 7 days. So, perhaps that sort of gives.
And even with ketones, it's true that in the animals, the ketones go up within an hour or two of fasting.
Wow. Whereas in humans, it's obviously dependent on exercise. 10 to 12, right? Yeah. So what I was sort of wanting to just touch on, for one, it seems as though intermittent fasting, in the context of certainly daily time restricted eating and perhaps even, doing a 24-hour fast or 48-hour fast, shouldn't have much of an effect in most women that aren't calorically restricting in addition to that, and perhaps aren't doing, running marathons or, running 15 miles a day, so there's obviously a spectrum here. When you're thinking about the effects on a woman's cycle, it seems as though it's the actual caloric load that's important. So if you're restricting your calories at the same time, it seems like it could affect.
That's right. Yeah.
But also in terms of just general benefits of intermittent fasting, in terms of the overlap between caloric restriction, it sounds as though there is some uncoupling. I don't know how much metabolic switching occurs with caloric restriction. Perhaps it depends on if you're doing something like a fasting mimicking diet where it's a pretty severe caloric restricted diet, or if you're doing something like these caloric restriction societies do.
It's very interesting. I don't know if have you met Rafa DeCabo?
I would love to have a conversation with him. I'm familiar with his work, but him and I have not. We were supposed to meet at a conference a couple of years ago, but that didn't work out.
So it turns out that most, perhaps all, of the rodent studies with rats and mice of calorie restriction are also intermittent fasting studies. And the reason is, the way they did the studies, they take 20 animals, you divide them into two groups, you, over a period of a week or two, determine how many food pellets each animal eats each day. Okay, then, sorry, you do that before you divide them into two groups. So essentially you get for each animal their daily calorie intake. And then you divide them into two groups, and the calorie restricted group, you give, say, 20% fewer pellets than they would have normally eaten each day. And you give it to them all at once. It turns out when that's done, because they're calorie restricted, they eat all of their food in a short time period, like within 4 to 6 hours. So they're actually fasting for up to 20 hours. So we did, I think it was the first study published where we asked, are there effects of intermittent fasting that are different or quantitatively greater or less with intermittent fasting and calorie restriction. So there's a certain strain of mouse that when we put them on every other day fasting, on the day they do have food, they eat pretty much twice as much food as they normally eat. And so over time, they don't lose any weight. Okay, so they're intermittent fasting, but no calorie restriction. And then we had calorie restriction group and another group we called pair feeding. But anyway, the bottom line is what we found is that we see in those studies is the brain. We saw clear beneficial effects of intermittent fasting on the brain that were independent of calorie intake. So we had animals that were on intermittent fasting, no change in calorie intake, and still saw. Actually, we did the epilepsy model and showed that it still protected them against epileptic seizures, protected the neurons. We still saw decreases of IGF-1. I can't remember all the details, but yeah. And then I mentioned the study with Michelle Harvey in England. That was really, we dissociated. Remember, we had a group that was counting calories each meal, and then we had group 5-2 intermittent fasting. And they both lost the same amount of body weight. And over 6 months, their calorie intake was the same. Yet the women on 5-2 intermittent fasting lost more belly fat and had a significantly greater improvement in insulin sensitivity. So that's, I think, the first human study to show at least some benefit that can't be accounted for by a reduction in calorie intake.
What about uncoupling benefits of weight loss from intermittent fasting?
We did that in the mouse study.
So we had the brain. Yeah.
Yeah. That study could be done in humans. You just have to. It hasn't been done.
I think there was a time-restricted eating study done by, was it Faraday? University of Chicago.
Krista Verity. Yeah.
Perhaps, I think there was no weight loss and there were some benefits. There was, metabolic benefits.
Yeah.
I can't recall all of them, but again, coming down to knowing what you've discussed with these stress response pathways and the cytoprotective mechanisms and, these, the adaptations and the metabolic switching and the importance of all that. What are your...
I'm going to have to leave pretty soon, but I want to mention one thing that's kind of interesting. So we found in rats, we implanted rats with essentially transmitters where we could record in real time 24/7 their heart rate and blood pressure. And then we switched them from ad lib to either daily 30% calorie restriction or every other day fasting. And we found that their heart rate and blood pressure went down over a period a couple of weeks, a little bit more down by a month, and then stayed down. Then we switched them back to ad libitum feeding, and at about 2 weeks, the resting heart rate and blood pressure going back up, and by a month, it was back to where it was way back at the beginning. So, a couple of things. So, people who do aerobic exercise tend to have low resting heart rate, low blood pressure, and increased heart rate variability. Heart rate variability is the variability in the time interval between individual heartbeats. So, for example, if your heart rate was 60 and mine was 60, that doesn't mean that each of us, there's every second, exactly every second, there's a beat. It could be 0.8 seconds, then 1.2 seconds, then 0.9, and then. So initially, I guess it's kind of counterintuitive that it's a good thing to have variability between than the time interval between beats, but actually it is. So endurance athletes have high heart rate variability, and essentially what it means is their heart regulation of their heart is more adaptable to change to stress and other changes. And so what happens is, and this has been known for exercise and particularly aerobic exercise, that the reason it causes a reduction in heart rate and blood pressure and increased heart rate variability is that the exercise over time will enhance what's called the parasympathetic nerves that innervate the heart. And the parasympathetic nerves slow down heart rate. The sympathetic nervous system increases heart rate. Okay, so we found the same thing with intermittent fasting. It enhances the parasympathetic nervous system. That will also, and that's through the vagus nerve, which is this big nerve coming down here. It innervates the heart, slows down heart rate. It can increase blood flow, dilation of blood vessels. And it can also enhance gut motility. So I guess I kind of want to throw that out there. It's kind of an interesting effect that I hadn't talked about of intermittent fasting on the cardiovascular system that's very similar to exercise. It takes a few weeks to a month to see the clear effects. Then if you stop doing it, you stop exercising, stop intermittent fasting, it doesn't take long for things to go back the way they were. And people who, like me, who had, you exercise regularly, and then in my case, for an accident and surgery, I had to stop exercising. So within a month, my blood pressure went from like 100/60 and resting heart rate of like low 50s to blood pressure like 135/85, and my resting heart rate's up like to 70. So yeah.
So
you got to stick with it if you can.
Yeah, I just want to mention sauna use real quick, because that's also been shown to do the same thing with parasympathetic activity and heart rate variability and blood pressure and all these things. And it mimics cardiovascular, or particularly aerobic exercise. And it's very useful for people that are injured and can't go out and get that exercise. It also helps maintain muscle mass. That's been shown in both animal and human studies. So keep that in mind, but also just... You should. And if you don't have access to a sauna, hot baths can also, and I can send you some of this information, hot baths can also increase heat shock proteins, which have been shown to help with preserving the muscle mass, and then heart rate. Your heart rate elevates.
Surprise.
Yeah.
Hot bath can increase heat shock protein.
Right. So, but I know you have to leave. I just wanted to ask you really two quick questions. One, you're just rapid fire thoughts on some of these so-called caloric restriction or fasting mimetics like resveratrol or spermidine or hydroxycitrate, things that have been shown to increase autophagy or polyphenols from coffee, and maybe doing that in combination with intermittent fasting and what your speculation would be, but also what your intermittent fasting routine looks like maybe before and after your injury, and if they're different, and then that's it.
Well, the answer to the last part is easy. Not different. I eat all my food within a 6-hour time window, and it's no simple sugars, virtually no saturated fat, mostly plant-based. nuts, vegetables, some fruits, fish. I do eat whole grains. I'm not so convinced that whole wheat is bad for you. I don't think I have any gluten. Okay, then.
Do you do prolonged fast ever, or longer than a daily times of eating?
Surprisingly, I haven't. And then about trying to mimic effects of fasting. We've worked on this a little bit with 2-deoxyglucose and with something called DNP, which is a mitochondrial uncoupler. 2-deoxyglucose is it's glucose that doesn't have a hydroxyl group that glucose does. That's why it's called 2-deoxyglucose. But anyway, 2-deoxyglucose will be taken up in cells just like glucose is, but it cannot be used to produce ATP, and it competes with an enzyme called hexokinase. That's like the first enzyme involved in the metabolism of glucose that leads to ATP production. So the bottom line is if you feed an animal or a human 2-deoxyglucose, cells in the animal will think, they will experience the effects of glucose deprivation because there's less glucose coming in. And in fact, it will increase some of these protein chaperones, one called GRP78, glucose-regulated protein 78. It's kind of like a heat shock protein. And we'd found that if we give 2-deoxyglucose every other day, it can be neuroprotective in some of our models. And we published that. And then with Don Ingram, not with Don Ingram, it was Don Ingram, he wanted to see if 2-deoxyglucose would increase lifespan. So he put it in the diet of animals, and actually it shortened their lifespan. It had some adverse effects long-term on the cardiovascular system. So now that's kind of mimicking calorie restriction at a kind of fundamental global way, way upstream. The things you mentioned are, can you activate pathways, like certain pathways, autophagy, and others that are maybe kind of downstream. And I don't know. I think the thing is, it's kind of tricky business, knowing how to know what amount of anything that mimics fasting is enough. For example, you may see some short-term benefit in whatever endpoint you're looking at. So, for example, ketones. The 2-deoxyglucose will increase ketone levels because the cells think there's less glucose in the blood when there's actually not, and so ketones are produced. But long term, it's not good. So, I'm kind of my advice is to stick with exercise. I think intermittent fasting can be helpful for a lot of people. At keeping your mind intellectually engaged, eating good diet. There's just not sufficient data to support the use of any of these things, whether it's nicotinamide, riboside, or rapamycin seems like it's something that in animal studies looks pretty interesting, but I'd be kind of hesitant to take rapamycin myself, just given that what it's prescribed for is suppressing the immune system, and long term in humans we don't know. So yeah.
I'm so sorry to have to ask you one more question, but I feel like we need another podcast. Just because you're so knowledgeable in this field, I would just love to know what your thoughts are. Do you think, for example, a type of, daily time restricted eating, maybe something similar to what you're doing, or eating you eat within a 6 hour window or 8 hour window every day, would have an effect on human lifespan, or at the very least health span?
I would predict it would. And this is in comparison to 3 meals a day plus snacks.
And what if the 3 meals a day plus snacks,
At
you're
the same
still
calorie
exercising
intake.
and healthy, and you're still metabolically healthy?
Say that again.
If let's say the person eating the 3 meals are eating high quality meals, and maybe eating some nuts or something for a snack, and they're also physically active, so they're lean and metabolically healthy.
I think maybe, because I don't know if that's true for humans, we don't know, but in animals, exercise alone without calorie restriction or intermittent fasting has minimal or next to no effect on lifespan, maybe like a 5% increase. So in animals, and in animals that are herbivores, calorie restriction, intermittent fasting have a striking ability to extend lifespan. Running wheel exercise, and the animals run quite a bit every day. Not much. But the animals don't die from cardiovascular disease typically, or diabetes. They die from cancers, kidney disease are kind of the two main causes of death. So in as much as exercise has really good effects on the cardiovascular system and glucose regulation, I wouldn't be comfortable extrapolating, the animal to the humans, because I think exercise is really important.
Yeah, if you look at the observational studies, exercise.
And my own personal experience is that the exercise for me has more profound beneficial effects, at least on my mental health, than intermittent fasting. And I think also on, I mentioned my blood pressure is now up. It's not like, high clinically, but it's high compared, so without exercise, even though I'm doing daily time restricted eating and my body weight's down, I'm still, showing these bad changes without the exercise. Yeah.
Yeah, right. Thank you. Well, I 100% agree. When I exercise, it's almost exclusively for the brain benefits and mood, for sure. And the other things are kind of healthy side effects for me. But I really want to thank you, Mark, for the discussion and just the wealth of knowledge and all the research you have done over the years. I mean, I have alluded to your research, just hundreds of times through public speaking and podcasts and YouTube videos and articles. And so, you've really had a major influence on my thinking, in the field of biological stress in general, and intermittent fasting, and the effects on the brain. And
so I've followed some of your podcasts and video casts over the years and enjoy them. I think you're doing a big service to the general public, and we need more people like you who are, presenting things, interviewing scientists and trying to present things and what's known, what's not known. There's a lot of garbage on the internet, and just like so you're beefing up the internet autophagy, maybe help clean up a little bit of the garbage. And
I appreciate that. I appreciate that a lot, Mark. And I know that you're in the process of writing a book, which is, waiting for your proofs back. So I look
Yeah,
forward
it's written.
to.
I'm done. I'm just waiting for the proofs. So,
I'm
and
looking forward to reading that and sharing it.
the second book is actually, in the general public, I'm kind of best known for work on intermittent fasting, but that's just a part of research over the years. So my second book is called Sculptor and Destroyer: The Story of Glutamate, the Brain's Most Important Neurotransmitter. My postdoc work, I showed glutamate played an important role in controlling the formation of synapses during brain development. At that time, in the 1980s, it wasn't recognized that neurotransmitters have an important role before their synapses form. It was thought, okay, the nervous system gets wired up, there's synapses, and now there's neurotransmitters. But yeah, so we showed glutamate plays an important role. And then, so that's the sculptor. And then glutamate's important in synaptic remodeling and learning and memory. So that's the sculpting part. Then the destroyer is, so I mentioned epilepsy and excitotoxicity. There's a lot of evidence that in Alzheimer's, Parkinson's, definitely stroke, traumatic brain injury, ALS, all of those, excitotoxicity is a factor. That is, neurons continuing to be excited by glutamate when they're in a compromised state energetically, for example, with a stroke, or we think more subtly in Parkinson's or Alzheimer's, where there's mitochondrial dysfunction that's occurring, and then the neurons continue to be excited even though the mitochondria aren't producing enough ATP to run the ion pumps that pump the sodium and potassium back. What was your PhD work was what?
My PhD work was mostly cancer metabolism, mitochondrial function. I did a lot on.
So in neurons, it's been estimated that in neurons that are active during normal activity, up to 50% of the ATP is used to drive the sodium pump and the calcium pump to pump those ions back out after the neuron is fired. So when the neuron fires an action potential, when it's active, the sodium and calcium rush in, then you get a voltage change across the membrane, and then that's propagated, and it's important to rapidly remove the sodium and potassium so that the charge across the membrane gets back to where it was. And so, yeah, during normal aging, Alzheimer's, Parkinson's, if the mitochondria aren't working even a little bit less well than they are, there's this tendency for hyperexcitability. And so anyway, that's the destroyer part of the sculptor and destroyer.
How much of a role would the GABA production from being in ketosis help negate that?
Well, that's important. That's a good question. I think that from a drug standpoint, drugs that enhance GABA tone could be beneficial. The trick is it's a tricky business because glutamate is critical for learning and memory, and it's all our circuits essentially are glutamatergic, so you don't want them to go out of control, but you don't want the activity to go too low so that function isn't optimal.
What about testing that with something like a ketone ester to see, transiently?
No, we've done that. So Richard Veach in I can't remember what year again. Anyway, bottom line is he sent a postdoc up to my lab, and what was his name? Kashi, a Japanese postdoc, and together with people in my lab, in a mouse model of Alzheimer's disease, they gave the animals in the food ketone ester or not, and then we had isocaloric, and then we looked at the amyloid accumulation, the neurofibrillary tangle, tau, and learning and memory, and the ketone ester was beneficial. And no, I think the ketone ester is very promising. A friend of mine up in Canada, Steve Cunan, has done some really nice work with PET imaging. So, all right. So one can get images of relative levels of utilization of glucose by brain cells or of ketones. So Steve Cunan he had radiolabeled glucose, 2-deoxyglucose, and radiolabeled, he used acetoacetate, but it doesn't matter. He didn't use beta hydroxybutyrate. Okay, so he had these were just initially normal people, but he's doing studies in people with mild cognitive impairment and Alzheimer's. So he had them, so when they're eating carbohydrates, brain cells use mainly glucose. When they go on a ketogenic diet, the brain cells switch. They clearly switch. It's very clear. They use a lot more ketones, maybe still using some glucose. And then in Alzheimer's disease, other investigators decades ago had shown that very early on, people with mild cognitive impairment, even somewhat during normal aging, there's reduced glucose utilization by brain cells. And however, we think, based on some of our animal studies and some preliminary studies in Alzheimer's patients of Steve's, that at least in people with early Alzheimer's disease, even though the brain cells have problems using glucose, they still seem to be using ketones very well. So this is something that is I'm excited about. And there's a neurologist that was under me at NIA that he's actually finishing up a study of intermittent fasting in people at risk for cognitive impairment. And he may be one of the places where these ketone ester studies are done. And then the why am I blanking on her name? Kieran Clark's work at Oxford with ketone ester and the elite British cyclists. Mm-hmm. So, yeah, that's a big deal in the endurance athletes, and the cost, right, for the average person, the cost of the ketone ester is way too much to make it practical, and we don't know for sure. It would be interesting to do like a comparison of someone fasting versus ketone ester. Then you could maybe sort out whether any endurance enhancing effect of intermittent fasting is in addition to ketone ester, or if it's all due to the ketones. But yeah, bottom line is, I think that's a really promising.
As you mentioned, the cost is very prohibitive for most people, and I think of it. So I can tell you from just anecdotally, and I'm so sorry. I know you have to go. It's great speaking with you. So as a sort of a foot in the door to like maybe someone who is not motivated to try a ketogenic diet, perhaps they have cognitive decline or dementia, or maybe even early stage Alzheimer's. When they notice a beneficial effect from the ketone ester, and that's a lot more motivating for a person to have a real world piece of evidence where they feel an effect and say, okay, well, maybe I can try this diet that's not that quite easy to do, but they may be more motivated
There's
to do it.
There's also, before he passed away, Richard Veitch, he interacted with, like, he had a friend who had Parkinson's disease, and he took the ketone ester and claimed that it had really clear beneficial effects on his Parkinson's symptoms, and that are fairly rapid. So,
thank you for telling me this. My father was diagnosed with Parkinson's.
People can try it. I mean, it's not going to hurt them. And
I'll tell you anecdotally. So two things. One, my father was diagnosed with Parkinson's about, I don't know, 3.5 years ago, and I have these ketone esters, and I've been sort of trying to get him to try it, and the pandemic happened and all this stuff, and so I'm going to try to go back to that point. But my mother has a different type of motor disorder. She has both orthostatic tremor and essential tremor. Orthostatic tremor is when she stands still, her legs will shake very rapidly. But if she walks, that doesn't happen. She's fine if she's moving. And the essential tremor is if she's like, eating or like has her arm out,
How old is she?
It'll shake. She is 63. She's had this. I mean, it pretty much started to go badly in after her menopause. She went through menopause, and all of a sudden, her orthostatic tremors got to the point where she couldn't stand. Whereas when I was growing up, she could stand without, her legs trembling so bad. So now it's like if she's in line, she has to have something to sit, where she can move fine, but like the standing.
And what about your grandparents? Did they have, any of them have Parkinson's or
No, not that we're aware of. No.
Okay. Well, that's so. Good.
But I was going to tell you I have convinced my mother to try the ketone ester, and it does improve both her orthostatic tremor and her essential. Mostly, it improves her essential tremor. But I'm using that as.
Did you do a blind study?
That is a great no. I need to do that. We've done it several times and it's worked, but it's hard because it tastes so bad that there's just no way she would not know that she's getting it. Yeah. She often has to chase it with something like a little bit of orange juice or something like that. So until the ketone esters, it is hard. So there's definitely a potential for a placebo effect, but there, knowing what we know about the epilepsy and seizures, and it seems very reasonable that it would help with motor dysfunction and certainly what we know about mitochondrial dysfunction. Yeah. So anyways, I've got these ketone esters, and I've been sort of trying to convince both of my parents to try a ketogenic diet. My father, though, he's actually sort of always naturally done some sort of intermittent fasting. Just that's sort of what he's naturally done, / maybe even a little bit of caloric restriction,
So he's around the same age?
He is 72.
Okay. Yeah.
And when
was he diagnosed?
When he was around, I would say 68.
Yeah.
About 68. Yeah. Mm-hmm. And so I've been, exercise and all these things in the lifestyle that I'm trying to like help optimize, but I really am wanting to see if there's an effect of the ketogenic diet. And I think the way to do that is to look at the ketone esters. That's how can you instantaneously put someone in ketosis without a lot of work? That would be it, So that's my little anecdotal.
Okay, Rhonda.
Cool. Well, anyways, yes. Thank you so much, Mark. I will let you get to your next thing you have to do. But can we please have another podcast and talk all about your destroyer and what was the second part? Sculptor. Thank you. I really enjoyed speaking with you. It's really great to have conversation with you all about science. So I'd love to do it again if you're willing.
Okay, and maybe your colleagues there that are doing this learned a little too.
Oh, I'm sure they're going to be super excited.
Okay.
So thanks so much, Mark.
Okay, bye, Rhonda.
Bye.